Hello again. We pursue our discussion and the questions and answers on molecular biology. And here you can see the results of Michelson and Stahl experiment. And I'll be going over this experiment at the end of the questions and answers. But now I'll be focusing on the importance of this experiment only. The importance of the experiment of Michelson and Stahl is to prove that the replication of DNA is semi-conservative replication. Because there were many theories about the types of the suggested models of DNA replication. There was the semi-conservative replication, of course, which is the result of the experiment, conservative replication, and dispersive replication. What's the difference between the two? Of course, the semi-conservative replication is the most accepted one, and there were proofs, experimental proofs for it, and that's why the hypothesis was the most accepted, and the other two theories were rejected. The dispersive theory suggests that the DNA molecule is dispersed, is broken down into fragments, and that these fragments reunite together to form random segments that form complementary strands. The conservative is the other extreme, which postulates that the two original strands are kept as they are, connected to each other via the hydrogen bonds, and meanwhile, while they are double-stranded and joined to each other, they form a new copy of them that is also double-stranded. But in the semi-conservative replication, there is separation of the two strands by breaking down the hydrogen bonds. And when the two original strands are separated, the new strands are built upon each of them, and therefore you end up by having two similar DNA molecules, because as I've just said in the first part, if in the original strand you had here A, adenine, then you had here thymine. And if you had here cytosine, then you had guanine. And if you had here thymine, again, then you had here adenine. So the original molecule was A base pairing with T, C base pairing with G, T base pairing with A, and when they separate and try to form new strands, each of them tries to form a new strand, it will form a complementary strand. So here we will have T, here we will have G, and here we will have A. Similarly, here we will have A, here C, and here T, and therefore AT is similar to AT of the gray molecule. Cg is similar to the original Cg of the gray molecule, and T gray and A blue is similar to the TA on the original molecule. And now we come to a question, or we move on to question number 14, and that question says, describe the process of semi-conservative replication, or what does semi-conservative replication refer to or mean? Semi-conservative replication. What does semi-conservative replication refer to? So we are going to mention three major steps or characteristics of semi-conservative replication. In semi-conservative replication, first, the two original strands are separated. The two original strands are separated. And then step number two, a new complementary strand, a new, complementary strand is formed on top, is formed complementary instead of on top of, is formed complementary to each of the original strands, to each of the original strands. This is step number 2. And the result is that we end up by having 2 DNA molecules that are formed, each consisting of, this is very important, of course, the 2 DNA molecules have 2 characteristics. First of all, the 2 DNA molecules are similar to each other. They are similar to each other, but meanwhile, similar to the original molecule and similar to the original molecule. So you had a parent molecule or a mother molecule, and then you had 2 daughter molecules that are similar to each other and similar to the original molecule of DNA that was trying to replicate. And then the fourth characteristic is that, so this is characteristic A or step A, B, and then C. And at the end of the day, we have that each of the formed molecule is formed of an old strand and a new strand that is complementary. So this is the end of the characteristics. So we have each of the 2 daughter molecules is actually a hybrid molecule. It is hybrid because here hybrid in the sense of one side is old and the one strand is old and the other is new, and they are complementary to each other. Of course, here a new complementary strand is formed complementary to each of the original strands. We would add according to the base pairing rules that we know, according to Watson and Crick base pairing rules that we all know. So this is about semi-conservative replication. And the next question, this is question 14, not question 4, question 14. We talked about ester bonds and dehydration synthesis, so I'll not be repeating it. And then here we have a very important question about complementary base pairing that appears frequently or is frequently encountered on tests. Question 15. Say whether the following is true or false. So you could have one of 2 formulas or similar formulas, like A, for example, plus G equals T plus C, or A plus G over T plus C equals 1. Or you might have another formula that is, for example, A plus T equals G plus C, and A plus T over G plus C equals 1. Let's first consider the one to the left. Of course, here, if the first statement, A plus G equals T plus C is correct, then definitely the second one will be correct. So because if A plus G, the left side of the equation equals the right side of the equation, then if you divide them by each other, then the resultant will be equal to 1. So is it true? Let's see what we know about the facts of complementary base pairing. We all know that A always base pairs with T via 2 hydrogen bonds. These are hydrogen bonds, not equal. So, and G always base pairs with C via a triple bond made of hydrogen bonds. But since A always bonds with T, then truly speaking, the number of bases of adenine bases is equal to the number of thymine bases, equal in number, equal in number. But when are they equal in number? They are equal in number in a double-stranded piece of DNA. Why in a double-stranded piece of DNA? Because it is the only way for base pairing. When we have one strand and the other strand, then if you have A here, then you have T here. So if you are considering the double strand, then this rule will apply, or this fact would be correct. But it cannot apply to a single strand, because you might have on a single strand 5 adenine bases, but you can have only 2 thymine bases. So in order for having the number of adenine bases being equal to the number of thymine bases, then it has to be in a double-stranded piece of DNA. And similarly, G equals C. Here, this is not the number of hydrogen bonds, but it is equality in number. So this is also true. So since in the first equation, A equals T, and in the second equation, G equals C, then we can deduce that by summing up the left side of the two equations, A plus G, the resultant will be equal to the sum of the right side of the two equations, which is T plus C. Of course, similarly, you can invert. That's to say, you can write, for example, that T equals A in number and that C equals G in number, or you can keep G like this, G equals C, and in such a case, you would have T plus G equals A plus C. The essential is that on the left side, whether on the left side of the equation or on the right side of the equation, the two values should not be purines or pyrimidines. You should have one pyrimidine base and another purine base. Yani in each or in whatever order, T plus G or G plus T. Similarly for the right side. So, but what cannot be correct is this. We cannot say that A plus T equals G plus C, because this necessitates that A, for example, equals G and T equals C. In such a case, yes, A plus T equals G plus C. But you can never guarantee, even in a double-stranded piece of DNA, that adenine equals guanine, because there is nothing to force them to be equal since they do not base pair with each other. And now we move on to question number 16. What is the evidence that makes the possibility of DNA being the genetic material, or if we want to formulate it in a simpler way, we would say justify DNA can be the hereditary material of the cell, the hereditary material of the cell. And now we will bring a series of evidence, pieces of evidence that DNA can be the genetic material. Number one, or DNA fits into being a genetic material or has all the characteristics that allow it to act as a genetic material. First of all, because having those nucleotides in different sequences, so we have different sequences of nucleotides, and therefore the genetic information can be stored in those sequences of nucleotides. For example, we can say then G, C, A, T, C, for example, is a particular gene. And therefore, if we have G, and then T, A, C, 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 this is a longer sequence. So this is gene 1 and this is gene 2. So since you have possible different sequences of nucleotides in different arrangements with different numbers and types of nitrogenous bases, possible different sequences of nucleotides, we will be using the term sequences frequently, then DNA can store the genetic information in those specific sequences. DNA can store genetic information. DNA can store genetic information in specific sequences or as specific sequences. So this is the main idea, that every specific sequence will represent a particular gene. The second evidence that supports that DNA, so here we are talking about evidence that supports that DNA is the genetic material. The second evidence is that every 3 consecutive nucleotides, consecutive in order, consecutive nucleotides in a queue, consecutive nucleotides. And of course, the most important are the nitrogenous bases, because it is the nitrogenous base that changes from one nucleotide to the other, while the ribose sugar and the phosphate radical, the phosphate group are constant. So every 3 consecutive nucleotides form what we call a triplet code. So what is a triplet code? A triplet code is a sequence of 3 consecutive nucleotides on a DNA strand. And that triplet code will represent or will be specific to a particular amino acid. So it will specify a particular amino acid, and we will see how during polypeptide synthesis, when the enzymes see, for example, a particular sequence like AUG, they know that they have to get the amino acid methionine to the ribosome in order to attach it or to start the synthesis of a polypeptide chain. The third characteristic that is very helpful is that the information can be transferred from DNA, can be transferred from DNA that always stays protected in the nucleus, cannot leave the nucleus because it might be destroyed by the cytosolic enzymes. The information can be transferred from DNA in the nucleus to messenger RNA, and messenger RNA is the delegate of DNA, and messenger RNA can then go to the cytosol and represent DNA or tell the cytosolic enzymes about the sequence of nucleotides on DNA, just because messenger RNA is complementary to one of the strands of DNA, as we will be seeing later on. So this possibility of flow of information from DNA to RNA is very important. And then the presence of this accurate base pairing, allows accurate replication of DNA strands, and therefore allows that the DNA material can be transferred from one generation to another without being lost or changed or mutated. And now we move on to question number 17. These are a bit more complicated questions concerning the wording of the question, but the answers are so simple and the content of the answer is very easy. So in question number 17, how does replication of DNA ensure continuity of genetic information? So the question is, see how the wording is a bit strange, is a bit unusual, but the content of the answer is very easy. So how does replication of DNA ensure continuity of genetic information, Number one, the first answer, we also have A, B, and C. So the first item in the answer is that there is semi-conservative replication that occurs, and therefore every daughter cell, before cell division, of course, I mean before mitosis, before mitotic cell division, or even before meiosis, the cell in the interphase undergoes semi-conservative replication. And semi-conservative replication that occurs before cell division, semi-conservative replication before cell division, and of course we know that it occurs in interphase, ensures that each daughter cell will receive an exact copy of genetic information, because during semi-conservative replication, an exact copy is formed, or two copies are formed out of the original molecule of DNA. So it ensures that each daughter cell receives an exact, and the word exact here is very important, an exact copy of genetic information, Second, what I've just said about messenger RNA. In the process of transcription, you have a strand of DNA that has a particular sequence, any sequence like A, T, C, G, A, for example, and that strand will be a template for a molecule of messenger RNA that will leave the nucleus and go to the cytosol to be translated into a polypeptide. So in front of T, we will have A. In front of C, we will have G. In front of G, we will have C. But complementary to A, we are supposed to have T. But since T does not figure on messenger RNA, we have the base instead that is also a pyrimidine, and that is called uracil. So this stretch of DNA has been transcribed into messenger RNA. So this process of building a strand of messenger RNA using the nucleotide sequence on DNA is called the process of transcription. So this is the process of transcription. And then messenger RNA leaves the nucleus to go to the cytosol to be translated into a polypeptide chain. And as I've just said, every 3 consecutive nucleotides are or represent one amino acid. So you have amino acid number 1, and then you have amino acid number 2. They are brought from the cytosol, of course, by enzymes that read the sequence on messenger RNA and bring the necessary amino acids to form the polypeptide chain, and this process is called translation. So what helped messenger RNA to be an exact delegate or representative of DNA? The fact that, again, there is complementary base pairing, meaning that when the enzymes see UUU, they know that on DNA we originally had AAA. So messenger RNA is a representative of DNA, and it is an accurate representative of DNA, and therefore the information is accurately transferred to the cytosol, again because of this accurate complementary base pairing. The third factor that ensures continuity of the genetic information, because when we say ensuring continuity of the genetic information, we are not only talking, when we say continuity of the genetic information, we are not only talking about the transfer of the genetic information, but we are also talking about the correct transfer of the genetic information, and this is ensured by the third process, which is proofreading, because the same enzyme that builds up a new strand of DNA, and that is called DNA polymerase, does what we call proofreading, meaning that if there is any wrong part of the strand of the new strand that has been built, then it is excised and removed, and then it is replaced again by DNA polymerase by a new correct sequence of DNA in order to prevent any mutation. So just like the actual building of a new strand of DNA, just like the actual building of a new strand of DNA is done by DNA polymerase, proofreading is also done by DNA polymerase. If there is an error in the sequence, in the new sequence, then there will be excision. This error is detected by DNA polymerase, and then excision occurs, cutting this error by endonucleases and exonucleases is done, and then DNA polymerase puts the correct sequence. So proofreading, and we will see this in detail later on, proofreading requires first that endonucleases and exonucleases act together. Endonucleases are enzymes that cut the DNA strand inside the molecule, not at the extremities, but the exonucleases cut the DNA sequences at the ends of or the extremities of the DNA molecules. This is what we call the process of excision. So proofreading requires first detection and excision, and then after that, DNA polymerase will build up the new strand of DNA in the same direction as we will see later on, which is 5' to 3'. And now we move on to the next question, which I think is question 17 or question 18. And that question says we will be talking about the process of DNA replication. And the question says directionality of DNA, directionality of DNA influences replication. Directionality of DNA influences replication. We have talked before about the directionality of DNA when we were talking about the DNA strands, and we said that for each strand we have at the end of the strand a free phosphate group, and since this free phosphate group is bonded to a 5 prime to the 5 prime carbon atom, then we call it the 5 prime end of the polynucleotide strand of DNA. And we also said that on the other hand, we have the 3 prime end, and the 3 prime end is the end at which we have a free hydroxyl group. So here at the other end, we will have a free hydroxyl group. And since the hydroxyl group is bonded to the carbon atom number 3 prime of the ribose sugar, then we call this end the 3 prime end. So what is the 5 prime end? It is the end at which we have a free phosphate group. And what do we mean by a free phosphate group? We mean a phosphate group that is not bonded to anything afterward. Similarly, we have the free hydroxyl group, and the free hydroxyl group is not attached to any other structure downward. But when we come to the double-stranded structure of DNA, we will find that the other strand of DNA will be in the opposite direction, meaning that we have here the 5 prime end and we have the 3 prime end. So if that strand has a free hydroxyl group here, so here we will have a free phosphate group for the red strand. And on the other hand, if we have here on this side uppermost part of the strand, we have a free phosphate group and a 5 prime end, then here we will have a free hydroxyl group and a 3 prime end, and they are connected, of course, by hydrogen bonds. So this is what we mean by the directionality of DNA. But in order, you can talk, of course, about the directionality of a single strand of DNA. This means that it has a 5 prime end where there is a free phosphate and a 3 prime end where there is a free hydroxyl. And you can also talk about the directionality of a DNA molecule. It's more difficult to explain the directionality of a DNA molecule, but we can say it in an easy way by saying that directionality of the DNA means that the free phosphate radicals, or the 5 prime end, the free phosphate groups, are at opposite ends of the molecules. The free phosphate groups are at opposite ends of the molecule. Or you might say similarly that the free hydroxyl groups are at opposite ends of the molecule, and this is what we mean by directionality of the DNA molecule. Sometimes we describe this directionality of the DNA molecule by saying that the two DNA strands inside a DNA molecule are anti-parallel. And it means the same thing. It means that the free phosphate groups are at opposite ends of the molecule, and that the free hydroxyl groups are at opposite ends of the molecule. Okay, so this is number one, what the word directionality mean. But how does this, the question is not to define directionality. The question is to say, how does directionality affect the formation of DNA molecule? So this is very important because we will say it now in a simple form, and then when we will know more about replication, we will have to add some information. So if you have, for example, this is the strand that the DNA polymerase will use in order, as a template, in order to build the new strand of DNA, then the new strand of DNA will be built in the direction 5 prime to 3 prime, meaning that the enzyme will build the new strand in this direction, because you know that if this is 3 prime, 5 prime should be here. It seems as if the other strand is upside down, and you can see it on the slides in the 3D picture of the DNA molecule. So this is the building up direction. This is the direction in which the DNA molecule will build up the new strand of DNA. So the blue strand of DNA is the new strand that is being built. And therefore, there is a famous statement that we always say that any new nucleotide that is added to increase the length of the strand will be added at the 3 prime end and not at the 5 prime end. So this is very important, that any new nucleotide is added at the 3 prime end. This is a very important piece of information that you frequently encounter in your test. So this new strand, or what we call the replicate of the original strand, you can say either that it is being built up in the direction 5 prime to 3 prime, or in other words, that any new nucleotide will be added to the 3 prime end. And now there is a very important diagram that is related to what we say and that we have to know. And that diagram will show you how we name the different strands of DNA and which one of them is used in order to to be transcribed into messenger RNA. So if you have the original strand, that is, for example, if you have one strand that is 5 prime to 3 prime, and that strand, of course, is made up of nucleotide, but I'm just representing the polynucleotide strand of DNA by a rectangle. But this means that the strand of DNA is led horizontally like this. And of course, the other strand would be like this, meaning that if this end is the 5 prime end having a free phosphate group, then this end would be the 3 prime end having a hydroxyl group. And this side, if it is 3 prime, then this side is definitely 5 prime. When we do transcription, meaning that when we use the sequence of nucleotides on DNA to form messenger RNA, we build the new strand of messenger RNA in the direction 5 prime to 3 prime. So building up is always in this direction, 5 prime to 3 prime. Then transcription should be onto this strand. We should use this strand to transcribe and not the other strand. Why? Because if you want your new strand messenger RNA to be built in that direction, so here you have 5 prime and here you have 3 prime, just because the enzyme of building RNA, which is RNA polymerase, can only build in this direction, then your template strand on which you are building should be complementary and should be in the opposite direction and should be anti-parallel to the messenger RNA that you want to transcribe. So this strand that is used for transcription is called the template strand. So what is the template strand? It is the strand that is being used for transcription. But that strand is also has two other different names. But before saying this, if this is the template strand, then the other strand will be called a non-template strand. But the templates, this non-template strand is not useless. This non-template strand is also called the sense strand or the coding strand, and I will tell you why. This is because this non-template strand carries the original genetic code as it is, and I'll give you the proof now or right away. Sense strand, so this one is called the anti-sense strand, and if this one is the coding strand, then that one is the non-coding strand. So it's all about understanding these two terms or the reason behind these two nomenclatures. Why is this one called the template strand, and why is that one called the coding strand? Afterward, everything would be understandable, because if this is the template, then definitely the complementary is non-template. And if this is coding, then definitely the complementary one will be non-coding. And we will understand why the coding is the sense strand. This one is called the template strand because it is the template on which you build up messenger RNA. So if here you have A, then here you will have U. And if here you have C, then here you have G. And if here you have G, then here you have C. So it is the template that decides the sequence of nitrogenous bases on messenger RNA based on the sequence on DNA, but of course following the rules of complementary base pairing. But if you look at that strand and at that one, if here you have A, then definitely here you had T. And if here you have C, then definitely here you had G. And if here you have G, then here definitely you had C. So if you compare that sequence with that sequence, you will find that they are similar. C, C, G, G, T, and the alternative of T on messenger RNA, U. So by having done this two-step process, having a complementary strand of DNA and then a complementary of the complementary would be similar to the original. And that's why this one is called the coding strand or the sense strand, because it is the one that will be represented by messenger RNA that is going to the cytosol representing the DNA and acting as a delegate of DNA. And now we're going to move on to question number 18 or 19, actually, question 19, which is, what are the proteins of replication? What are the proteins used in replication? What are the proteins used in replication? Actually, the proteins used in replication are not only enzymes, but they are enzymes and non-enzyme proteins. So first of all, we would have the helicase enzyme. And the helicase enzyme would be preceded by a topoisomerase enzyme. Topoisomerase enzyme. And I don't mean preceded in time, but I mean ahead of it on the DNA molecule. And number 3, we would have a third enzyme that will do the actual building of DNA, or actually two enzymes. We have a DNA primase, and then we will have a DNA polymerase, and then finally, in order to join the Okazaki fragments that we will be talking about later on, we have the DNA ligase enzyme. So these are 5 enzymes, but in addition to those 5 enzymes, we have several other proteins that are used in replication, of course, of DNA, but the one we will be discussing is the single-stranded binding protein. Single-stranded, it's a protein made of a single polypeptide strand, single-stranded binding protein. I would like to emphasize that concerning the repair that we have talked about before concerning exonucleases that remove the incorrect segments from DNA at the extremities and endonucleases, they are not only responsible for excision, but they are responsible for detection of the incorrect part. So if I had said any other thing, if I had said that it's the polymerase that detects, it's incorrect. I'm sorry. So detecting the damaged parts of DNA and excising them is the function of the endo and exonucleases. And now we will see how all these enzymes work together in order to cause DNA or to help the process of DNA. So this is just mentioning the proteins used in DNA replication. And then the next question would be to describe the function of each of them. So question number 20, describe the function of. I would discuss the function of helicase enzyme together with the function of the topoisomerase enzyme, And I'll tell you why. But actually, if you have a double-stranded DNA molecule like this and another double-stranded DNA molecule like this, and the molecule is supposed to continue here. So in eukaryotes, I'm talking about eukaryotes, then there will be a site where the replication or like a level point where the replication will start, and this is what we call the origin of replication. So the origin of replication is the site on the DNA molecule where the replication will begin, and because the because the DNA molecule is very long, then we cannot do this one shot, so we divide it into segments. And at the beginning, we will have two helicase enzymes that will be acting in opposite directions. So there will be a helicase enzyme that will start here and another helicase enzyme that will start there. What do the helicase enzymes do? They walk along the DNA molecule in opposite directions. And what allows them to move in opposite directions is that they start inside the DNA molecule. They do not start at the periphery. They start in the middle of the DNA molecule and walk in opposite directions. What do they do when they walk? Say, for example, we call this helicase 1 and that one helicase 2. What do they do? They, first of all, unwind the DNA molecule, meaning that they convert they change the DNA shape from being 3D and in the form of a helix, and they convert it into a ladder-shaped structure. And then after having done this, they break down the hydrogen bonds between the complementary base pairs. And when the hydrogen bonds between the base pairs are broken down, then the two strands are kept apart. But we do not do this along the whole length of the DNA molecule, but in this segment only. So when the two strands separate in this strand only, we obtain something that looks like a bubble, and we call this a replication bubble. We call this a replication bubble. And each half of the replication bubble is called, together with the rest of the straightforward strand, a replication fork. Because look at this structure, together with the straightened part of the DNA molecule, it looks like a fork. So every replication bubble consists of two replication forks, and this is the result of the action of the DNA helicase enzyme. As we said, there should be unwinding first of the DNA molecule helix, because sometimes the helix is too tight. So in front of the DNA helicase, there is another, or ahead of it, there is another enzyme called topoisomerase. Topoisomerase does what? If helicase breaks down the hydrogen bonds, but the enzyme that walks ahead of it, and that is called topoisomerase, will not act on the hydrogen bonds. It will act on the sugar phosphate backbone, on this main part of the strand of the DNA, and break down the covalent bonds if the turns or the coils of the DNA molecule are too tight, in order to enlighten the tightness or diminish the tightness. So if there weren't a topoisomerase enzyme walking ahead of the helicase enzyme wouldn't be able to perfectly unwind the two strands of the DNA molecule, and then to break down the hydrogen bonds in between them. An additional thing that we might add here is the single-stranded binding protein. What do the single-stranded binding protein do? The single-stranded binding protein bind to the separated DNA molecules, to the separated DNA strands, in order to prevent them from rejoining together. So this was about the action of the DNA helicase, about the action of the topoisomerase, and about the actions of the single-stranded binding proteins. And now we will move on to question 21, where we will be talking about the function of the very important DNA polymerase. And we said a very important enzyme because it is the DNA polymerase that does the actual building up of the DNA molecule, the actual building up of the DNA molecule. So how does DNA do this? Of course, there are certain properties for the DNA molecule. So again, the question is, describe the function of DNA polymerase. Describe the function or functions of the DNA polymerase. Of course, there are rules for the DNA polymerase. The first rule for the DNA polymerase is that it adds new nucleotides. And this is how you should describe the function. It adds new nucleotides to the 3' end of the newly formed strand, or of the replicate, or of the new strand. And therefore, this means that the new strand is built up, in the direction 5' to 3', in the direction 5' to 3'. And number 3, the third characteristic is that it follows the base pairing rules of Watson and Crick. So it follows Watson and Crick base pairing rules. It follows Watson and Crick base pairing rules. adenine binds to thymine via 2 hydrogen bonds, and guanine to cytosine via 3 hydrogen bonds. And the fourth characteristic is that it builds up new strands for the two original strands of DNA. One of them will be built one shot, and the other strand will be built in segments. So, for example, if this is the replication bubble, and if this is the direction 3 prime 5 prime for the original strands, and 3 prime 5 prime to the other original strand, and if this is the replication bubble, and if this is the level of the origin of replication, then it will build the first strand. When it is still active and still energetic, it will build this strand in one shot. So, for example, it will build it like this. So it would be, of course, complementary to that one. And this would be building up in the direction 5 prime to 3 prime, because even if we take a segment here, then that side is 3 prime and that side is 5 prime. This one is similar to that one, and this one is similar to that one, because it is the same direction. So if you build a new strand on top of this strand, then it will be built one shot. And that strand that has been used as a template will be called the leading strand. So it will build up the complementary to the leading strand in the direction 5 prime to 3 prime as usual, but it would be done one shot without rest. Whereas the other strand would be built up again in the direction 5 prime to 3 prime, and we'll be explaining this in full detail on the second screen. But because this segment has been built up into fragments, and because it is built up after the first one, so we call it the lagging strand. So the leading strand is built up one shot, whereas the lagging strand is built up in segments that are called Okazaki fragments, and we will be explaining this on the next slide, which is a continuation of the answer of question number 21. So here we have the original DNA strands, okay? So we would say that this is the 3 prime end, and this is the 5 prime end. And as I said, if to the right this is 3 prime, meaning that I have a free OH, and to the left this is 5 prime, meaning that I have a free hydroxyl, a free phosphate, then if you take any segment inside this, then you will always have the OHs pointing here and the phosphates pointing there. So at any segment inside the molecule, in the middle of the molecule, this side will be the 3 prime end, and that side will be similar to here, the 5 prime end. But because this is the 3 prime end, then this is definitely on the other strand the 5 prime end, and here the 3 prime end. So if this is the origin of replication, then the leading strand will be built one shot like this in the direction that is opposite. So if this right side is 5 prime, then on the built new strand it will be 3 prime, and if that side on the original, this is the template strand, of course, this is the template strand, this is the original nomenclature, it is the template for replication, then this would be the complementary, which is 5 prime. But because this strand has been built up in one shot, so it is called the leading strand. So don't be confused that this is the leading strand, the strand that is newly formed in the direction 5 prime to 3 prime in one shot, whereas this one would be built in segments. These segments are called Okazaki fragments, And that's why the other strand, because it's built up in segments, in Okazaki fragments, then it is called the lagging strand. But of course, this strand will also be the template strand for the Okazaki fragments. And of course, again, each of the Okazaki fragments will be built up in the usual direction, which is 5' to 3'. And then comes question number 2. How are these Okazaki fragments ligated together? Because the Okazaki fragments cannot stay like this separated from each other. So we should have a DNA ligase enzyme. And at this level, this is the only function you have to know about the DNA ligase enzyme, which is to join the segments of Okazaki fragments together in the process of DNA replication. But later on, you will find that you will know and learn that the ligase enzymes are also used for repair of DNA. And now comes the remaining enzyme, which is the primase enzyme, or the DNA primase enzyme. This is the remaining enzyme, and question number 23 would be about the function of the DNA primase enzyme. DNA polymerase, the enzyme that is responsible for walking on the template strand and gathering the complementary bases to form a leading strand, cannot start from scratch. There should be a sequence of DNA that is pre-made and that is ready here. And the enzyme starts building by adding the first nucleotide to this pre-made sequence, which is called the DNA primer. So the DNA primase function is to form a DNA primer, and a DNA primer is a short sequence of DNA that's built at the origin of replication to allow the DNA polymerase enzyme to start its work in building up the new strand by attaching the first nucleotide to the 3' end. Why have I said 3' end? Because I'm talking only about this segment that also starts 5' and ends 3'. So the the presence of a well-functioning DNA primase enzyme is important to allow the DNA polymerase to to start its work, and we will be now drawing a sequence of nucleotides on the template strand. The template strand is always in the direction 3' to 5', just like the new strand is always built in the direction 5' to 3'. So question number 23 is, how is the DNA primer built? How is the DNA primer built or built up? So if you have a sequence like this where you have C, T, A, G, C, C, T, G, C, A, T, C, C, for example, then the primer will be built in the middle. So it will start being built here at the T. So the primer is built, the primer is the DNA primer, of course, because there is RNA, there is what we call RNA primer. So the DNA primer will be built or is built starting here. The DNA primer is built starting here. Then how is it done? It will be built in the direction 5' to 3'. So if this is the 3' end, of course, because we are to the left, and this is the 5' end, then the primer, and I'll draw the nitrogenous bases or the nucleotides of the primer in red. So the primer will be built in this direction, complementary to this part of the DNA strand. So here you will have A, and then in front of the G, you will have C, and then G, and then T, and then T, and then A, G, G, and it would be built, it would, in this way, it would have been built in the direction 5', which is complementary to 3', and here 3' complementary to 5'. This is the DNA primer, and this is how it is built. And now when the DNA polymerase comes to start building its new strand on top of this template strand of DNA, it will start here. And this is a suitable starting point for DNA polymerase because it can only bind new nucleotides to a free 3' end or an end where we have a free hydroxyl group. So this is again how the DNA primer is being built. So if we draw again the diagram of the replication bubble, and this would be question 24, to show the position of the DNA primers on the DNA molecule, because in a replication bubble, you will have two DNA primers. So we will have a leading strand like this, or a template strand like this, and here another template strand, and here we have the hydrogen bonds in between that haven't yet been broken by the DNA helicase. Suppose that here we have the 5' end, then here we will have the 3' end, and then therefore this will be the 3' end, and that will be the 5' end. Of course, in both cases, this is called the template strand. This is the strand that we build on, and this will be the template strand for the molecule, that the strand that will be formed on top of it. And this will be another template strand for the other, because we are talking about semi-conservative replication. So each of the strands will be used as a template. But if we consider that this is the origin of replication, if we consider that this is the origin of replication, then we will divide the replication bubble into two halves. And for each half, then we will have here a leading strand and its corresponding lagging strand. And similarly, on the other hand, the leading strand will be in the opposite, on the opposite direction, meaning that here you would have the leading strand, and here you will have the Okazaki fragments. So here, this is the leading strand for template number 2. And this is the lagging strand that is formed on template number 1. And on the other hand, here is the leading strand that is formed on template 1. This is the leading strand on template number 1, and this is the lagging strand for template number 2. So at the end of the day, every strand of DNA molecule that is complementary to one of the template strands is formed of a leading part and a lagging part. Where are the primers located? The primers are at the beginning of the leading strand. So here we will have a starting primer, a DNA primer, a DNA primer, and here we would have another DNA primer. So at each origin of replication, where we start the process of replication, we will have a DNA primer. The DNA primer is built in the direction 5' to 3', and similarly here, it is built in the direction 5' to 3'. And then, as we said, the Okazaki fragments will will join together the DNA strands. The question now, question number 24, is that why do we need the DNA polymerase to turn and replicate the other strand in the form of lagging strands? Why doesn't the DNA polymerase, while walking along, suppose that this is the molecule of DNA polymerase, why doesn't it add complementary nucleotides on each side, and that's it? Why to do it on one side and then turn and do it on the other side and be a bit tired and exhausted and do the other side in the form of lagging strand? This is again because the DNA polymerase should build the new strand in the direction 5' to 3'. If it tries to build the two strands at the same time, then the first strand would be accurately built in the direction 5' to 3', but the second one would be built in the direction 3' to 5'. So the question is, justify. Question 24, I think, if I remember. Question 24. So you have a justify question. DNA polymerase does not build the new strands at the same time. Does not build the new strands of DNA at the same time. There is one that is leading and that occurs first, and another that is lagging and that occurs afterward. This is because the answer is DNA polymerase has to build. The simplest answer is because DNA polymerase has to build each of the two new strands, in the direction 5' to 3'. So if it built them at the same time, which doesn't happen, if it built them at the same time, one would be built in the direction 5' to 3', and the other would be built in the incorrect direction, which is 3' to 5'. By having said this, we would have reached the end of this video, and we would have covered most of the important information in part 1 of the molecular basis of inheritance. Thank you.